Hi there, thank you. Thank you, Kevin, for the introduction, and to Warren for organizing this event. I'm from the Malaria Atlas Project. We make pretty maps of where malaria is. And as Kevin said, I'm going to be concentrating on Plasmodium vivax, which has been my focus for the last several years, and how we have been mapping and addressing the challenge of this relatively neglected parasite. And because we have a strict 8 minutes to cover what we need to cover, I'm going to do a flying tour of some of the key points about Plasmodium vivax, malaria biology and epidemiology, why it's considered neglected or how, what challenges we've faced in trying to map this disease, and what we've done to try to meet that challenge. So as Kevin mentioned, there's 5 or 6, if you count the subspecies of ovale, parasites that affect humans for malaria. And the vast majority of the burden or global caseload is attributed to falciparum and vivax malaria. The biology of Plasmodium vivax does impart some unique epidemiological problems that make it difficult to control and even to understand in some ways. As shown here in this life cycle illustration of Plasmodium vivax, you'll notice that there's this liver stage. All Plasmodium parasites do enter the human liver at some point in their life cycle. But ovale and vivax have this ability to stay in the liver and form this dormant phase called the hypnozoite. That stage can then wake up at some point. It could be anywhere from a few weeks to months to years after the initial mosquito bite primary infection, and cause another blood stage infection and then another one after that. And this ability for it to sort of hide out in the liver allows it to overwinter seasonal transmission. It allows it to hide from blood stage treatment, diagnosis. And if we look at a map of what malaria looked like at the turn of the previous century, you see that it extended far into the temperate climate. And that would have been Plasmodium vivax, that light green way up north. And it remains the most geographically widespread of the human malaria parasites. It's still far up in North Korea. And an additional problem of this relapse, so not only does it hide out, but it's difficult to treat that liver stage. There's one currently licensed drug that can treat it, and it comes with its own problems, which could be a whole nother discussion on its own. But it's a long treatment schedule, which people don't like to take a drug for 2 weeks. And it can cause acute hemolysis in people who have an enzyme deficiency called G6PD. So some people are reluctant to give it as a drug. There's other things that Plasmodium vivax does that makes it hard to find. It circulates the blood in low parasite densities. It's often submicroscopic. It's often asymptomatic as well, so people don't necessarily have the classic malaria fever to go for treatment. So we have this widespread parasite. It's hard to understand. It's hard to find. So why would this be one that's considered neglected? And I've been told by a couple reviewers of papers that we can't really call vivax neglected anymore. The fact that I'm even invited to here to talk about it, I think, means that it's gotten some attention. But when we talk about it being neglected, it's relative to falciparum, because as Kevin said, when we talk about malaria, most people think falciparum, Africa. And just to kind of test this hypothesis, I did a search on PubMed of falciparum plus malaria, around 30,000 hits. Then vivax plus malaria, only around 7,000. So it is quite clear that there is more research output for falciparum malaria. And within MAP, our own database that we've assembled of cross-sectional surveys that look for infection in communities, we have Plasmodium vivax here. This is a map of all of our survey points. And we have around 13,000 points, which I was quite proud of because I was, one of the people going around basically begging people for data, and was feeling quite proud of that until I compare it to falciparum, which we have about 42,000 points for. So there's a huge discrepancy even within our own group where we use the same methods for both parasites that we don't have as much evidence to support what we try to do for both. So given what we have available and what we know about these parasites, what could we do in terms of trying to map it and understand how differences in the spatial distribution of this? So what we started doing at MAP was the first thing we did several years ago was we mapped the limits, the geographic limits of transmission, and that allowed us to assess what the population at risk is worldwide. And then within that, we estimated the prevalence using those survey points I showed on the previous slide. But what we're now trying to do is generate an estimate of how many new clinical cases happen each year, which is a very different metric, but we think is more operationally useful for people who work within country programs. So in order to go from prevalence to an incidence map, we first needed to understand, does relapse vary geographically? So this phenomenon of when the hypnozoite wakes up. Because there's been a long-standing hypothesis that the tropical strains will wake up quite quickly after the primary infection, whereas the temperate strains wake up more slowly so that they last through the non-transmission season. And so we did a large analysis and found that there are indeed geographic zones of relapse. So we developed a mathematical model to relate prevalence, which we had mapped, to incidence, which we want to map, and we had separate model fits for those different geographic zones of relapse, assuming that relapse would affect both prevalence and incidence measures. But our models and our outputs are only as good as the data that we have to put into that. So if you look on the previous slide, some of our relapse zones have almost no data, and some of the zones had no data. And I was fitting this to about 175 points. And these 13,000 points that I have as our evidence to predict prevalence, you can see that they're not homogeneously spread out throughout the Vivax endemic world. We're heavily biased to areas that have national household surveys like DHS and MIS, which don't commonly search for Vivax, and from areas where we have support from other Oxford institutions where we have strong collaboration. So by far, most of our data is from Indonesia, thanks to our colleagues based at the Eichmann Oxford unit. So what can we do to meet this challenge? We don't have a lot of data. We have a complicated parasite. And what we want to do is capitalize on the type of data that is strongest from different regions of the Vivax endemic world. So in the Americas, they have strong, relatively strong health systems, health information systems. So we could use incidence data that is reported from health facilities to the government to measure incidence. And in other areas where we know that the government isn't reporting any Vivax, like in Africa, we can use this modeled relationship that we have. And the outcome is that we have a hybrid map that utilizes health information system data, which we of course adjust for things like reporting completeness and treatment seeking, because we know that even in strong health information systems, not all the facilities report, and not everyone who gets sick goes for treatment. But even with those sort of flaws that we have to adjust for, we still think that an adjusted health information system database is better than a model that has a weak evidence base. As things progress towards elimination and strong control, the irony is that the less malaria an area has, the better that they are at finding their malaria cases. And we're finding that in some elimination, the countries that have already set elimination goals, that they're able to find, record, and geoposition cases better, such that we can make more fine-scale resolution maps and allow for malaria stratification for control and elimination subnationally. We only have a handful of countries, but I'm very optimistic that this is going to improve even further over the next 15 years, as Kevin showed with the funding slide. So that's a very brief overview of what we've done with Vivax and MAP, and I want to thank our whole team that's contributed in different ways, and our funders, the Wellcome Trust and Bill and Melinda Gates Foundation.
Okay, thanks very much. So I'm going to turn that over to the panel. I don't know who wants to take the first one. The first question was about interactions between falciparum and Vivax, and one could either talk about that, well, biologically, geographically, treatment-wise, any way one wanted. So, Katherine, I don't know if you want to go first with that?
Sure. Of course. Is this close enough to hear?
I think so. Let us know if you can't hear. It sounds to me as if it's good, yeah.
So as far as mapping the two parasites, we've kept them very much separate just because of the methods that we use have been different so far. But it's definitely something that as we get into this kind of fine scale mapping that I mentioned on my very last slide, it's becoming more important. But what we've seen as far as our data that's available is that where they're co-endemic, Vivax doesn't tend to get picked up as often. So there's some diagnostic biases to it, but then there's also some of the biology where, and then I'm probably not the best to speak to this, but where it's simply not detected, even if it is there. So it's something that we're considering how best to represent this in our maps to show, especially because of the importance of mixed infections potentially on clinical outcomes. But as of yet, that's something that we're still working on as far as how to find these mixed infections and how to show them in the maps and how important they are.
Great. Nick, do you want to, or does anyone else want to comment? I wondered if you want to comment on the biology, as it were, the interactions within a single individual, some of the sort of stuff that you've been considering.
Okay, so these two parasites are actually in competition with each other, and we know that because in the good old days when we used to treat neurosyphilis with malaria by giving malaria to people, people actually did that. They gave the two together, and you could see that one would suppress the other, and usually it's the muscular falciparum that pushes down the rather effete Vivax. I actually think relapse may be a strategy to avoid falciparum, but in Southeast Asia, where Katherine has shown you you have very frequent relapses, the two commonly coexist, and interestingly, falciparum malaria is an important cause of Vivax malaria. How does that work? Well, about 1/3 of people who get acute falciparum malaria will have a relapse of Vivax afterwards. So that, if you add that up, that means that falciparum as a stimulus, if you like, to Vivax relapse becomes very important. So these two are coexisting and trying to avoid each other, and as Katherine points out again, Vivax tends to be underestimated. So as falciparum comes down, you start to see Vivax. It's always been there, but you underestimated it initially.
So the next question was about chloroquine resistance in Vivax. Perhaps you'd like to start it, then maybe fill in if you leave anything out.
So yeah, chloroquine resistance is very interesting. We haven't actually nailed chloroquine resistance. We don't understand the molecular mechanism as we do for falciparum malaria. It's insidiously spreading. You get high level chloroquine resistance in Vivax in Indonesia, on the island of Sumatra, and throughout the Indonesian archipelago, and particularly on the island of New Guinea, so Papua New Guinea and West Papua. Elsewhere it's low-grade resistance, but it's increasing, so increasing late failure rates. In fact, they're probably failure to suppress the first relapse. As a result of that, the latest WHO guidelines do recommend now ACTs as an alternative, and that allows, it's very good, it allows you to have a unified treatment policy, and so ACTs are recommended. One caveat, Vivax develops resistance to pyrimethamine very rapidly, so not sulfadoxine pyrimethamine, but all the other ACTs can be used, and so probably chloroquine is gradually going to phase out.
Philippe, did he miss anything out?
Well, maybe just a comment. There's a lot less data on Vivax than on falciparum, and we are starting an effort of pulling data on Vivax, and this is a work which is led by Rick Price, And this is just the beginning of that. But when we look at the data from what we have seen already, the challenge of assessing this data is going to be higher than on falciparum. So Vivax is a complex beast, and it's going to take probably more research and more data collection to get to the understanding of that. One last thing that we are observing in Africa, for instance, like in Angola, we have seen a massive increase, and in Ethiopia, a massive increase of Vivax in proportion of falciparum in the last decade. As we see falciparum declining, we see the proportion of Vivax increasing. And so this problem, which was sort of only thought to be in Asia and Latin America, is likely to be also a problem in Africa at big scales in not too long.
Okay, so anyone else want to comment on the Vivax-falciparum interaction? If not, let's move to the third question, which was what's driving resistance from that one small part of Southeast Asia? Dominic, do you want to have a go at that? I mean, I've heard you venture views on this.
Well, I don't know.
Yes, that was the view you ventured last time, yeah.
I mean, I'm not necessarily the best to express all the vast range of different views that have been put forward. But clearly, some of it is possibly to do with history of drug use, and these drugs were introduced early on, and there were some rather dubious policies for administering intramural drugs over the years, so there may be partly historical issues. Partly it's to do with patterns of drug use. There's a lot of drugs being used, but also it's a region of relatively low transmission compared to Africa. And that means to say that many cases are symptomatic and many cases get treatment. Whereas in Africa, of course, many people are asymptomatic and never get treatment. So if you're in the parasite population, mostly in Africa, you're not getting hit by drugs. Whereas if you're a parasite in Southeast Asia, if you think of it as a game of whack-a-mole, the parasite's getting whacked more often in Southeast Asia. So that could be another factor. It's also plain that the parasites, these tribal groups that I talked about, have got some very interesting features about them, and it looks like maybe some of those features make it easier for resistance to evolve in those particular tribes. But I'm sure that Nick's got lots of other possible reasons.
I'd just like to say that in the large trials that have been conducted to map artemisium resistance, led by Dr. Liz Ashley, who's hiding in the corner there, the parasites were made available to anybody who wished, any bona fide investigator who wished to investigate them. So there are a lot of artemisium resistant, well-characterized parasites available for researchers should they want them.
Yeah, thanks. Well, while people are still cogitating about it, I'm going to ask Dominic to start on that, and then anybody else who wants to come in, because, well, simply because I know some of the work that's going on in Dominic's group.
Well, as you know, malaria is transmitted by a whole range of different Anopheles species. And as you know, in Africa it's dominated by some major species groups, Anopheles gambiae complex, for example, Anopheles fenestus. And as you know, in Africa, they tend to bite indoors in general. And so it's turned out that vector control in Africa has been a tractable problem. And now Nick, I know, can speak to this much better than I can, but in Southeast Asia the problem is there's a much greater diversity of vectors. They're often biting outdoors. They often, in a village there'll be many Anopheles species and no one's quite clear which are transmitting malaria, and they may be transmitting it in the jungle, etc. So vector control, relatively speaking, has been less of a powerful tool in Southeast Asia. Is that fair to say?
Absolutely.
So that's really the big that's the big difference. That's the sort of big thing that's going on in the vector. Now, and as you know, in the maps that we saw of the control of malaria that's going on in Africa right now, the vast, most important thing in reducing malaria mortality in Africa over the last 15 years has been the use of insecticides, insecticide-treated bed nets, indoor residual spraying, and as you know, insecticide resistance is now sweeping across Africa. So what's going to happen there is unclear. There's a lot of talk about using gene drive systems, etc., potentially new insecticides, but many of the issues that we've heard talked about in relation to drug resistance and vaccine development apply also to the mosquitoes. Is it going to work? Is it going to be sustainable? How do we guard against new forms of resistance? So there's a huge host of problems there, and obviously we think since from a genomic perspective, that can somehow help in classifying some of these problems, but they're very practical problems in the field.
Do you want to say, you mentioned gene drive, do you want to say just a little bit about that? Just people may not be so aware of the kind of sort of wild ideas that people are playing with now.
Well, it's been speculated for now for quite a long time that there could be genetically modified mosquitoes, and that could work in a number of different ways. The field has really sort of taken off. Everyone's got very excited about it in the last year or two because of a very efficient system for modifying genomes known as CRISPR-Cas. It allows you to edit genomes extremely effectively, and now everyone is thinking that this will be the way forward for modifying mosquitoes. On the other hand, it turns out we've been genome sequencing mosquitoes across Africa, and it turns out that thus far in Anopheles gambiae we find one variation every 2 bases. So firstly, finding a sequence that's sufficiently constant to get a gene drive system in, there's a limited number of sites you can target, and there's the potential that the mosquito population will adapt. So there's probably no magic bullets, but there's a lot of excitement about potential new approaches.
First question about whether, a mixed question, ACT resistance less likely in Africa because of the relative less use of monotherapy, and also some more consideration to the idea of triple therapy, which Nick mentioned in his talk. Nick, I guess we should put this one to you.
Yeah, I think there's several reasons why artemisinin resistance emergence would be less likely in Africa, but unfortunately it hasn't been possible to prevent monotherapies being used, despite WHO's, I think, quite forward-looking and brave ban. It was many years ago that the former head of the Global Malaria Program, Arata Kochi, said ban monotherapies. But unfortunately it hasn't been possible to really make it work everywhere. Still, I think for that, because of the higher transmission setting, it is probably less likely. The more difficult one, perhaps Kevin would like to comment, is whether we can persuade people, generally, to use 3 drugs, which means more adverse effects and more costs. We managed to do it for TB, we managed to do it for HIV, but malaria is a remarkably conservative grouping, and I don't know whether we can in malaria.
Anybody else want to comment on it? Yes, Philippe. And then
Yeah,
maybe also slip into the second question as well, was whether we can get to near elimination without resistance. I guess, sort of finally, it's almost like an arms race.
Well, first a comment on monotherapy and the quality of antimalarials also circulating in Africa. That's a piece of work that we are doing in WARN and led by Paul Newton. And we have been observing a very, very large amount of poor quality medicine, not only falsified or counterfeit medicine, but poor quality medicine circulating in many endemic regions, in Asia, in Africa, and in Latin America. We don't quantify that very well because there's very scarce data, but each time that it has been looked at it seriously, that's really striking a level of poor quality medicine circulating, and that's exactly what you would do in a lab. You give a subtherapeutic level of a treatment, and then you get resistance. And this is happening. This has happened, and this is happening today in Africa because of a lack of a capacity to really measure the quality of medicine. And despite the ban of artemisinin in Africa, and this has been working progressively better. There's a lot of poor quality ACT circulating in Africa, and we don't really talk about that. It's a sensitive subject, difficult to measure, but also sensitive because this is also touching the generic companies, and we don't want to affect the huge impact that has been in access of the generic companies. But on the other hand, we don't tackle properly, I think, at this point, poor quality medicine.
Great, thanks. So let's take, and does anyone want to comment on that question, which I took to mean there being something of an arms race, as it were, but can you get to elimination without resistance just becoming the absolute, barrier to it? Philippe, do you want to comment on that?
Yes, if you're quick.
I'm not sure I want to comment on that. elimination without resistance. Well, there have been some good results already in some part of Africa where we see a massive decrease of cases of symptomatic malaria. Now, are we really effective? I'm not sure. When we are looking now with much more sensitive methods than microscopy, like PCR techniques, and we see very striking level of parasitemia in patients in, let's say, Dakar, in an urban area where supposedly there's no transmission. Well, we find 20% of people having some parasites circulating in their blood. And so I think unless we have proper methods and proper ways of getting to this reservoir, I think we are a long way away from elimination.
Nick?
I think it's a matter of how quickly and effectively you do it. If you do it very quickly and very effectively, yes. And that's why we've been making such a fuss about artemisinin resistance. We know that the longer you drag it out, the more selective pressure you're providing and the higher grade resistance will happen and the partner drugs will fall like dominoes, and that's exactly, I'm afraid, what happened. I mean, we've known about artemisinin resistance in 2007. So Sri Lanka, for example, has eliminated now. They've eliminated without resistance.
Okay, let's just take the final question, then we'll take some more questions from the audience. And this was the effect of climate change, the potential effect of climate change. Will we be able to start doing our malaria research in Holland and the Fens again, as we used to be able to do? Katherine, I guess this falls more naturally in the mapping group, and certainly some of your colleagues have speculated on this.
Yeah, this is something that we've looked at in MAP with how we've looked at trends over time. And the change in climate that we've seen happening that could drive up malaria is luckily not as quick or effective as all the efforts that are going in currently to control malaria. So our control is more effective than climate change. I mean, there still are some epidemics in a warm period in, say, the Kenyan highlands that happened in the late 1990s. But overall, we're doing a better job at controlling it. What I think is probably going to be more of a problem than climate change is changes in land use. And this is what we see a lot in Asia, again, in this problem area where we see resistance, is we also see deforestation, which creates different environments and can allow breeding sites and changes in transmission. And I think that's kind of the thing to keep an eye on, is how we're using and changing the land.
While we are passing the mic, I'd like to comment on the AMFM. An interesting outcome of AMFM, which has not been really noticed. One of our colleagues at the London School of Hygiene and Tropical Medicine did a very interesting assessment of the quality of medicine in Nigeria. And they found in one of the states 13%, 13% of poor quality medicine and almost no falsified medicine in their very good survey in 2000, which was conducted in 2010-11. And we were sort of very surprised by that, and they say, well, you know, maybe the problem of poor quality medicine is not that big in Nigeria. And when they presented these results, there was somebody from the Global Fund who was part of the AMFM effort at the time, and she said something very interesting. She said, well, that was also the year where AMFM was scaling up in Nigeria, and we were pouring Nigeria of good quality medicine. And I think that was a very, actually, good demonstration that if you made available good quality medicine, you pushed away poor quality medicine and falsified medicine.
Yep, it's an important point, although correlation isn't causation, as we're often reminded, but well, it is because it is just undoing sort of all your sort of lecturing to students. Sumi, you've been sitting patiently waiting for a vaccine question. Here's the big moment.
I think firstly I would like to say it is a very important milestone, the efficacy results of RTSS. It's a vaccine against a parasitic disease that has shown any efficacy, so that is good. But as you point out, the efficacy is very modest, and there are all the likely implications of short-term efficacy, which will probably shift the malaria incidence into older population. And it is effective, and a lot of mathematical models have suggested it is cost effective and it averts case, etc. But I think what we are currently doing will be very informative. That is to deploy it, first of all, to see logistically if it is possible to deploy this vaccine, because it requires 4 doses as opposed to 3, which we can fit into the extended program of immunization, but also looking at a lot of other indications over time whether this vaccine will be effective in controlling malaria. So as you raised, I think these are things that we definitely need to look for and measure, but also keeping in mind that it has conferred some protection against a parasite that has been very difficult to achieve.
Okay, great. So as we enter that sort of important few minutes before we all go out for the reception, I want to just ask each member of the panel in 30 seconds or so to comment on the fact that in the Twitter sphere today, those of Nick, I suppose you've been on it all day, there's an enormous amount of sort of naive optimism about eradication and such like. We all want to eradicate malaria. But I want to ask each panel member to say how, on Malaria Day, when the sort of the word has been ending malaria, how optimistic are you that malaria can be ended in that sort of relatively short time span that people are talking about? And what would make you more optimistic? What one change would make you more optimistic? So we don't need a big discussion, just want to know, are you optimistic or not? But what I really want to know is what one change would make you more optimistic? Dominic.
Oh. Well, I mean, I think it's a disease that can be eradicated, and I know it won't get eradicated unless people want to eradicate it, so setting a timeline is good as long as it's not detrimental to other things. So I'm completely behind it, and whether it's 25 years or 50 years or whatever, we have to have a timeline and a roadmap. We have to be realistic. I think that the one thing would be realism.
Okay.
Just,
yeah, that's great. Philippe, optimistic or not, and what one thing?
Well, I think we need to learn at polio, possibly as an example, and we see how painful it has been to get where we are on polio. I think it's unlikely that the world will go back to another exercise like that, and we will need probably to integrate, so that's the positive note, we'll need to integrate the malaria effort with other efforts that are in public health that are facing those countries. Unless we do that, I think this is going to be a waste of time.
Okay. Nick, you optimistic?
I was optimistic about 10 years ago. I've become less optimistic. I agree with Dominic. I would say honesty. If there was actual honesty, true honesty in the dialogue, so that we did talk about things like corruption and bureaucracy and ineffective national malaria control programs, things we're not allowed to say, then I think we would have a better chance.
Okay. Simi.
Yes, I am optimistic, and the one thing that would make a difference, and I'm partially biased, is an effective vaccine, and I think that has to be used with all the other tools that we currently have, and one day we will get there.
Great. Finally, Katherine.
Cautiously optimistic, I would say. I think when we talk about eradication and getting rid of all malarias, when we consider things like nosy, that's where we have to be cautious. But I think when we look at the progress that we've made with PF and PV, that this is a possibility. And my one thing would definitely be a Vivax planted, a safe, effective treatment for the hypnozoite that people want to take. I think could get rid of this long-standing parasite of PV.